ければと思っております。はい。私自身はですね、2月にこのライデンに訪問して、トキシでちょっと話をしてきました。で、今回のセミナーは、基本的になんていうんだろう、非営利というか、技術的なプレゼンっていう形になっているんですけども、彼らも、やっぱり営利企業なんで、日本でのプレゼンス高めたいよということがあるので、今日皆さんとですね、画面、今日ウェビナーという形で、あるんですけれども、この後どんどんやり取りをしていただければというふうに思ってます。で、必要だったら私を介して言ってください。で、今日はですね、多くの方々に登録をいただいておりまして、製薬、それから化学屋さん、農薬屋さん、さらにはですね、政府の方、それから当局の方、で、アカデミックの方からも参加をいただいているので、非常にもし、聴講者としてもばらけているのかなというふうに思っております。じゃあちょっと私の方はですね、ここで、ストップする形にしたくて、Can I introduce your team? So, the first speaker will be Paula Van Rossum, and she's a CBO of Toxys, and she did a lot of liaison stuff before she joining, and now acting as a BD and sales to promote Toxys, the portfolios. And she will be the first speaker, and followed by Dr. Hill, spelling Gill, Hendricks. Sorry, it's my pronunciation. And he's the CEO and the founder of Toxys. And he did from basic research and now growing so much bigger for the companies. So he will do the technical bit of a presentation. So if I can hand over to Paul, first of all.
Okay, good morning and konnichiwa. Thank you very much for joining us today. First, to set the scene a little bit, there are a few trends that I think are very important in Europe and in America. It is really about moving away from animal testing. And so here is just a few recent pieces of news from the European Committee, FDA, EPA, and basically all of these big regulatory bodies are sharing that they want to really move away from animal testing. It will not happen in an instance because a lot of scientific proof is still needed, but ultimately everything will move towards really kind of phasing out animal testing. And I think this is also something that that drives us as a company, because we believe that the future of human safety assessment is not only with very simple assays that provide yes or no answers or animal models, but is really in the in vitro models that provide relevant insight into the mode of action. Now, of course, this is not only these regulatory bodies, but actually a number of industries are already actively moving towards using these new approach methods. And here are some examples with references, reference publications. The first one is a case study for the pharmaceutical industry, where they really integrated new approach method in assessing developmental toxicity hazards. Another example here is for using these types of assays for fragrance materials. And here is an entire strategy, NGRA strategy, next generation risk assessment strategy for the cosmetics industry. And of course, the cosmetics industry has no choice but to test without the use of animals, at least in Europe and in America. So this is basically why we exist. And so the question is, if we want to move away from animal testing, why do we want to do it now, right? And I think animal testing, it takes a long time. It is quite expensive. It's ethically debatable. So there are some drawbacks there that you want to overcome. NAMs will inevitably, as said, become part of the regulatory domain in many industries. But also at this moment, NAMs already help you to make better decisions. So why start now is really to already capture the value from these NAMs, have better understanding, making better decisions. and also building up your knowledge and your expertise of understanding what kind of value these assays bring. Because as I said, inevitably, they will become part of the regulatory frameworks. So as said, this is why we as Toxys as a company exist. On one hand, we're a biotech company, so we continuously develop novel in vitro assays, always focused on can we understand the mechanisms of toxicity. And on the other hand, we offer some of these assays as a CRO, but around the world.
Paola, Gil, can you hear me?
Okay. So this is a short overview of a unique portfolio. So you can see we have all of the green assays basically here. Our assays we really developed ourselves. I will not go into the depths of the technical details of these assays as that will be part of Giel's presentation. But so you get a little bit of an overview of all the things we can help you with. And these assays, as said, they're really kind of broadly adopted. We work globally with the top 10 pharma companies. We work with all of the biggest cosmetics companies and agrochemical companies. But we also have some global license agreements, for example, with Charles River, with LabCorp, with IVS, that offer ToxTracker. And our data is also widely used in all sorts of regulatory submissions to FDA, to EMA, ECHA, EPA, EFSA. So it's really quite widely used across the industries. And Giel will in his presentation show you a few examples on how these assays have been applied across different industries. So with that, thank you so much, and I'll hand over to Giel.
Thank you, Paula. And thank you, Masashi, for inviting us to give this presentation. I will try to share my screen. Here we go. So, good afternoon. As Masashi said, so my name is Giel Hendricks. I'm the CEO and founder of Toxys. Today it's a pleasure to share something about our technologies and how they are applied. And I will give you an overview of some of the in vitro models that we have developed for chemical safety assessment of pharmaceutical chemicals and cosmetics ingredients. And if we're talking about safety testing, then ultimately the question is which model is fit for purpose. So what model will give us the answer about the safety of compounds to humans. And this can be from very simple 2D cell culture models or more complex systems like organoids, organ on a chip, all the way to the actual animal models that will represent humans. But every time the question is, what is the best assay to give us the answer to predict the effects in humans? How do we capture the complex biology that is relevant for our question? How do we capture things like metabolization of drugs in the liver, for instance? How do we capture immune responses or maybe hormonal changes? And at the same time, also have a look at the costs, the reproducibility of the assays, the availability of the models. And the question is actually, do we still need animals to protect humans? In some cases, I think it is, the answer will be yes. But in some cases, I think science is really right to actually move away from animal testing. And FDA is in the US is pushing for a change, trying to move away from animal testing and at least opening the regulations to support also other methods. The International Council for Harmonization, ICH, is pushing their guidelines to accept also in vitro models as an alternative to animal testing. There's workshops organized by ECHA, by EFSA here in Europe, also by EPA and FDA in America, to create roadmaps to move away from animal testing, or at least to allow also new methods, new or non-animal models to be accepted. And there's also funding available to actually make this happen. So one of the areas where there's definitely a huge demand for alternatives to animal testing, it is testing for developmental or reproductive toxicity. About 50% of all animals used for safety testing are used for DART, and having, let's say, cheaper and faster options available to at least do part of the safety testing would be highly appreciated. And over the years, definitely a number of assays have been developed by industry, by regulatory agencies, often using human stem cells, the human iPS cells that were developed in Japan originally, as a surrogate for early embryonic development, often combined with in silico predictions. And the advantages of these models are that they're obviously much faster, they are cheaper than in vivo testing. They also can provide additional information on the mechanism of action of a chemical. They would obviously contribute to a reduction of animal testing, but there's also limitations. Of course, it's a simplified biological system. The exposure duration is much shorter, and we're still looking at interspecies differences. So at Toxys, a number of years ago, also we took the challenge to start developing an in vitro model that could capture the early embryonic development and looking at the biology in this stage from the, let me laser pointer, after fertilization of the oocytes, looking at early development of the fertilized egg into a blastocyst, and ultimately early development or differentiation of the stem cells into the primordial germ cell layers, ectoderm, mesoderm, endoderm, which would then ultimately lead to the formation of a wide range of organs in the developing embryo. And we reason that if we can develop a model that can capture this early process, we can use that to predict early developmental toxicity. And this ultimately led to the development of an assay that we call ReproTracker. ReproTracker is a cell-based assay that is using these human-induced pluripotent stem cells that we in vitro differentiate initially into the early embryonic lineages, mesoderm, endoderm, ectoderm, and then drive differentiation into more mature tissues, cardiomyocytes, hepatocytes, and neural tissues. And by looking at changes in morphology, by looking at the functionality of the differentiated tissues, and also by looking at changes in expression of a selection of biomarker genes, we can visualize that early embryonic development process. So what does it actually look like? So for cardiomyocyte differentiation, we see these pluripotent stem cells, and in a course of 14 days, we drive the differentiation by adding different growth factors, changing medium into cardiomyocytes that at day 14 start to express structural proteins, in this example, troponin T2, that are seen in cardiomyocytes. The cells actually start to contract, mimicking beating cardiomyocytes. And by looking over time at changes in gene expression, we see, by looking at this pluripotency gene, we see the loss of pluripotency of these cultures. Around day 3 to 7, we see expression of mesoderm biomarker and BMP4. And upon maturation, the expression is going down again. And at the same time, we see the induction of early cardiomyocytes and also late cardiomyocyte biomarkers. So in this way, we can visualize and follow the differentiation process. And we can do the same for liver. So this over a course of 3 weeks, we drive the differentiation of these cells into hepatocytes. It actually starts to look like hepatocytes. They start to express albumin, liver-specific transcription factors. And again, we see the loss of pluripotency, early expression of endoderm markers, and then upon maturation, expression of hepatocyte-like or hepatocyte-specific genes. And without any surprise, we also do this for neural differentiation, driving the cells into neural rosettes in 2 weeks, again, expressing neural-specific genes. And also we can, in time, we can follow the changes in gene expression for those selected biomarkers. So what does it actually look like? So as an example, the mother of all teratogens, thalidomide, which was given in the 50s to pregnant women to treat morning sickness, but later on clearly induced all kind of birth defects and the limb malformations. If we treat the differentiating cells or the Ripper tracker assay with thalidomide, then on top you can see the effects on the biomarker expression. So for cardiomyocytes, the mesoderm differentiation doesn't seem to be affected, but clearly at later differentiation we completely lose the proper formation of cardiomyocytes, as seen by the loss of expression of this biomarker gene, myosin heavy chain 6. We also see a loss of contraction of the exposed cultures. The morphology is disrupted. Also for the liver, we already see for liver that endoderm differentiation is affected, and clearly there's really no proper liver differentiation. We see that the morphology is also disrupted, and this by microscopy. Interestingly, the neural differentiation was not affected at all. So there's clearly some specific mechanisms at play here. And for thalidomide, now we actually know what the molecular mechanism is, but I don't have the time to go into the details there. Based on these changes, we would classify thalidomide as a teratogen. But also, of course, not everything is a teratogen. So as an example, cetirizine is a histamine 1 receptor inhibitor. So it's given to people with to suppress allergic reactions. If we treat the Ripper tracker cells with cetirizine, there's no effect on mesoderm formation. There's no disruption of expression of cardiomyocytes. You could even see a slight increase. There's no effect on contraction. The morphology is normal, and the same holds true for the liver as well as the neural differentiation. So based on the lack of effects in these cells, cetirizine is then classified as a non-teratogen. So to use this assay in a regulatory setting, a number of years ago, ICH, a guideline, mental toxicity and reproductive toxicity testing, ICH. Rat or rabbit was affected, then the in vivo call will be that it's a teratogenic agent. And so we compared it for the vast majority. We saw that the Ripper tracker classification correlated very well with the in vivo results, although there were a few differences. For the in vivo negative compounds or non-teratogenic substances, for nearly all of the cases, Ripper tracker also classified these substances as negative. But there's a few differences. Sometimes we believe that this is caused by simply species differences. One of the challenges here that we are using human cells to predict the effects in animals. And in some cases, we know that animals respond differently than humans. So there's simply a lack of human data to do this comparison. But for now, we simply said so we will consider these differences as either false negatives or false positives. And then when we calculate the accuracy of the assay, we get to about 85%. So in 85% of the cases, the in vitro model correctly predicts the results in rats and or rabbits. And it is definitely higher than any of the in vitro models that are currently available on the market. So we're continuing the validation of this assay and looking at species differences and improving the protocols to see if we can increase the accuracy a bit further. Already, this assay is used by companies in their testing strategy. One example is the pharmaceutical conglomerate, I have to say, Medicines for Malaria Venture. So it's a collaboration with a number of pharmaceutical companies to develop novel medicines for malaria treatments. And MMV is using ReproTracker as an early screening tool to drive their DART testing strategy, where if a substance is positive in ReproTracker, they would accelerate a red EFD study. So they would fast track that. If it's negative, they would continue with the development of the compound, and then at some point do an in vivo test that is obligatory. The qualification of ReproTracker would allow MMV to replace or to use ReproTracker as an alternative to the second species. So a red study in combination with ReproTracker would be sufficient in that case. In Europe, some of the companies, some of the industries, specifically the cosmetics industry, are not allowed to use any in vivo models. So in collaboration with Unilever, we have been working on what they call a next generation risk assessment framework. So an in vitro only based safety assessment of cosmetics ingredients, and also their ReproTracker is used as one of the assays in their in vitro testing strategy for developmental toxicity. And in collaboration with a number of partners, we're really trying to work on a pipeline that would allow the implementation of these new methods, combining PBPK models and in silico prediction tools, follow up by in vitro tools like ReproTracker, but there is also maybe a few alternatives to capture the biology in an in vitro model. Add mechanistic information from other assays like omics technologies to really build a case and understand or predict whether substances are toxic to humans. And following the ICH guidelines, this would definitely be a replacement for the second animal species that are required for DART testing. Okay, then I would like to switch gears and close off the DART part, because another area where Toxys is really active is in genetic toxicology. And genetic toxicology is definitely an area where in vitro testing is already generally applied. So where testing for DART heavily relies on animal testing. The available guidelines across the world, in US, in Europe, also in Japan, already include in vitro testing for genetic toxicology, where in the in vitro phase, typically using the Ames test and the micronucleus assay, compounds are screened for their mutagenic potential, as well as for induction of chromosomal aberrations or clustergenicity, as well as numerical chromosome changes or aneuploidy. And then a follow-up in vivo testing the same endpoints, typically using the transgenic rodent assay or PIG-A for mutations, and the micronucleus assay, maybe in vivo comet assay for chromosomal aberrations. So I said this is the battery of in vitro assay that we're currently using. So it's the Ames test for mutations, when positive, typically followed up by mouse lymphoma assay or an HPRT gene mutation assay, in combination with the in vitro micronucleus assay, and sometimes also chromosomal aberration assay. But I think this is becoming less. And then depending on the industry that you are working in, at least this is a picture that is currently used in Europe. For cosmetics industry, after an in vitro test, there will be no in vivo follow-ups possible, actually. For chemicals, depending on the production volume, in vitro testing is sufficient if all negative, or sometimes requires in vivo follow-up. And on the pharmaceutical side, there's no way to get around in vivo testing, where you have two options, either to use 2 in vitro models followed by 1 in vivo, or 1 in vitro model followed by 2 in vivo tests. The challenge is that if you look at the results from the in vitro as well as the in vivo assays, that quite often the in vitro models do not predict well the outcome in vivo. The Ames test is sometimes produces negatives for substances that are definitely mutagenic in vivo, but in many cases the in vitro models produce positive results that are not translating or not predicting correctly the results in vivo. And this is especially true for the in vitro micronucleus assay. And because there's no additional information coming from these assays, it's very difficult to translate positive in vitro to in vivo. And this is already also recognized, for instance, in a publication by the European Center for Alternatives to Animal Testing, or ECVAM, where there is simply already a list of chemicals that are non-DNA reactive that should give a negative result in an in vitro mammalian cell gene tox assay, but have been reported to be positive either in a TK mutation assay, so mouse lymphoma assay, or a micronucleus assay. So it's also recognized by the regulatory agencies that the in vitro models sometimes produce misleading positives. And so to improve that situation, a number of years ago, we have developed ToxTracker. ToxTracker is a stem cell-based reporter assay that are genetically engineered to express 6 different fluorescent reporter genes that together can very accurately predict genotoxicity in vivo. I will introduce the biomarkers on the next slide. The assay is able to discriminate between direct DNA reactivity and secondary or indirect genotoxic effects that is coming from oxidative stress or protein damage. We can use the assay to discriminate between classogenic and an angiogenic mode of action. And overall, by combining these biomarkers, the assay is able to provide insight into the mode of action of genotoxic substances. This assay is widely used as a screening tool for industry, as a follow-up to typically to a positive in vitro positive result, to explore the mode of action. And assay is often used as part of a regulatory submission. So I said the assay contains 6 different fluorescent reporter genes that detect different direct and indirect genotoxic effects. There are 2 biomarkers that are activated upon the induction of DNA damage. There's a biomarker that is activated by the induction of mutagenic DNA lesions, so bulky DNA adducts that actually block the DNA replication fork. It's dependent on ATR check1 signaling. We have a biomarker that is activated by the formation of DNA double-strand breaks, so classogenic DNA lesions linked to the ATM NF-kappa B signaling pathway. We look at activation of the p53 tumor suppressor response. And then we have 2 biomarkers that look for induction of oxidative stress, so the formation of reactive oxygen species that can indirectly cause DNA damage, as well as protein unfolding, so the activation of the unfolded protein response that will typically trigger an apoptotic response, triggering chromosome fragmentation. And it's a combination of those biomarkers that is telling us in a quantitative way whether genotoxicity is a direct or a secondary indirect effect. But to give you an indication how, or an idea how that assay works, I would like to share a small video. It will run about 3 minutes to give you an impression. What does this assay look like?
ドッグストラッカーとは、革新的なステムセルに基づいたレポーター圧生です。薬物やその他の化合物の遺伝毒性及び発がん性を迅速かつ確実に同定できます。まず、各化合物について広い範囲で細胞毒性の容量試験を行い、ドッグストラッカーで試験する濃度を決定します。1日目に96ウェルのプレートにワイルドタイプのステムセルを播種します。2日目に危険物質を DMSO または水に溶解します。続いて、危険物質を倍々で20段階の希釈を行い、S9 核分ありとなしの条件下でステムセルに暴露させます。暴露して24時間後に危険物質処置群と非処置群において細胞数を測定し、細胞の生存率を確認します。50% から 60% の細胞死をもたらす濃度をドッグストラッカーの最高濃度として採用します。通常、一番低い濃度では細胞毒性を示しません。この予備試験により、化合物の溶解度及び自家傾向に関する情報も得られます。次に、化合物を6種類のドッグストラッカーセルラインにかけ、DNA 損傷、 B53 活性化、酸化ストレス及びタンパク質ダメージの弱気程度を評価します。化合物を5種類の濃度で S9 核分ありとなしの条件にてレポーター細胞に暴露させます。暴露して24時間後、フローサイトメトリーにより6種類の GFP レポーターの誘導及び細胞生存率を測定します。独自のトックスプロットソフトウェアを用いて、トックストラッカーレポーターの差値的活性化を同定します。これは化合物の作用メカニズムに関する詳細な知見を提供します。最後に試験結果を詳細な報告書にまとめます。お客様のご希望に応じて、データの解釈や作用メカニズムのさらなる検討に関する情報を提供させていただきます。
So as you've seen, the assay is using flow cytometry to measure the activations of different fluorescent reporter genes. So we can measure GFP signals on a single cell level. And this is just a typical example of a readout of ToxTracker. So these are 4 in vitro genotoxic substances, but you can immediately appreciate that the responses in the assay are completely different. So the DNA cross-linking agent cisplatin on the left clearly induces in red the 2 DNA damage reporters, indicating that there's DNA adducts formed that block DNA replication, there's DNA strand breakage, there's p53 activation. So signs of a DNA reactive substance. The tubulin poison Taxol only induces strand breaks, but there's no DNA adducts formed. And arsenite gives a lot of oxidative stress, some protein reactivity, but no DNA damage detection. And as well as tunicomycin that triggers the unfolded protein response. And this is very relevant because cisplatin is the only compound that induces mutations in the Ames test, but all of these 4 compounds are causing pauses in the in vitro micronucleus assay, but they clearly have a different toxicity profile. So if we do the calculation, how well does this predict in vivo genotoxicity? Then ToxTracker has an accuracy of over 90% in predicting in vivo gene tox, and therefore does a much better job than the current in vitro models that we're using to predict genotoxicity. Understanding the mechanism is very important also from a regulatory point of view. This is just a statement from a publication from the European Food Safety Agency, where they clearly state that there is a general consensus that genotoxic agents acting via a non-DNA reactive mechanism exhibit a threshold non-linear dose response, meaning that there's a safety window for non-DNA reactive genotoxins. Where if compounds directly bind to the DNA, there's no safety threshold. So it has immediate regulatory implications. Over the years, we've expanded the protocol for ToxTracker to capture some additional mechanisms. One of them is to discriminate between clastogenic and an angiogenic mode of action. Again, angiogens being an indirect genotoxic effect, having a different regulatory importance. To discriminate between clastogens and angiogens, we use the power of those DNA reporter genes, where we get activation of both reporters or typically only one of the 2 reporters. But we combine that then with analysis of the cell cycle, looking for blocking of cells in mitosis, which is typically seen for these angiogens. And then after 24 hours, we actually measure the DNA content in the cells. And we can nicely see for these angiogenic agents that we see cells with an abnormal DNA content, aneuploid or polyploid cells. So in this way, we can also use the assay to discriminate between the DNA reactive clastogenic effects or angiogens, typically compounds that either induce microtubule disruption or inhibit cell cycle kinases. The other important mechanism to discriminate for genetic toxicology is secondary gene tox related to oxidative stress. And we can look, we can investigate this in ToxTracker by adding so-called Ross scavengers, chemicals that bind to the reactive oxygen species. And if we do that, then we can nicely see that in the case for the DNA methylating agent MMS, where we see oxidative stress as well as DNA damage being induced, if we add N-acetylcysteine to capture the reactive oxygen species, we lose the oxidative stress response, but there's still the same DNA damage response seen, suggesting that MMS is directly binding to the DNA, where potassium bromate, where we also see a lot of oxidative stress as well as DNA damage. If we suppress the oxidative stress in this case, we also lose the DNA damage response, suggesting that this, in this case, gene tox is a secondary effect going back, meaning that there's a threshold mechanism here. So to allow ToxTracker to be used in the regulatory space, we have started a formal OECD validation a number of years ago, where we have been involved in a inter-laboratory validation according to OECD guidance document 34 to validate the assay for regulatory applications and ultimately to work towards an OECD test guideline for ToxTracker. This is a project that has been overseen by an independent validation management team. We had 7 laboratories running the assay independently. And basically the goals of this study was to validate the assay again to predict genotoxicity, to look at the intra and interlaboratory reproducibility, and investigate whether that mechanistic information from that assay is useful. And the assay was performed in 3 phases: in installation, proficiency testing, and ultimately the actual validation. These were the partners that were involved. So the project was overseen by a validation management team led by David Kirkland. Toxys was only there as a supplier for technology, but was not really involved in the validation process itself. And then here you see the 7 laboratories that actually perform the validation. So companies from pharma, cosmetics, chemical, as well as 2 CROs. Just to give you an indication, just an idea, this is an example of etoposide, a DNA damaging agent that is tested at 3 labs, and it's clear, although there are some slight differences, clear that the responses are very, very similar across the different labs, showing immediately the reproducibility of the assay in different laboratories. When we looked at the reproducibility within the labs and between labs, we saw that within lab reproducibility was very high, maybe with the exception of 1 lab that was slightly underperforming, but overall anyway, anywhere between 80 and 97% within lab reproducibility. And even if we include that slightly underperforming lab, we still noticed a between lab reproducibility of over 83%. And again, when we calculated the predictivity or the accuracy to predict genotoxicity, we saw a sensitivity and specificity close to 90%. And just to give you one example, we also looked in whether the mechanistic information from the assay had additional value. And this is an example for 2 compounds that are negative in the Ames test, but positive in the in vitro micronucleus assay, but are generally considered to be non-genotoxic in vivo. And then if we look at the results from ToxTracker, it was clear that these compounds give a very strong oxidative stress response that is clearly the primary mode of action. If you push the system far enough, at some point you start to see some DNA strand breakage, but clearly these substances will be classified as non-genotoxic in ToxTracker, and actually thereby correctly predicting the in vivo outcome. Okay, in the last 5-7 minutes of this presentation, I would like to introduce a 3rd assay. This assay is not widely used for regulatory applications, but is definitely a valuable screening tool for pharma, but also for chemical read across for the chemical and maybe also cosmetics industries. And this is an assay that we call Tox Profiler. It's a HepG2 based reporter models, again, genetically engineered cell lines that express a wide range of fluorescent reporter genes, capturing, again, things like oxidative stress, cell death, cell proliferation, but also induction of inflammation, protein stress, ion stress, autophagy, stress to the endoplasmic reticulum, and inhibition of cell cycle progression. This assay is not using flow cytometry, but high content microscopy, allowing us to not only look at changes in expression or induction of the genes, but also at changes in localization, which indicate activation of a protein, translocation to the nucleus, for instance, or the formation of these intracellular foci. And by measuring the different responses of these different biomarkers, we can, in a very quantitative way, see what is the driving toxicological effect for a certain compound. And from these microscopy pictures, we can derive these dose response plots where, in this case, for 4 different chemicals, we can see whether there's a preferential induction of either oxidative stress. Cadmium chloride induces ion stress, also some protein damage. Mitomycin C, the DNA damaging agent, blocks the cell cycle primarily. Verapamil induces a lot of autophagy. So in a quantitative way, we can measure the toxicological effects of a substance. And we can use this in a kind of a heat map approach to capture the point of departures to see what is now the primary toxic response that a compound gives. And we can also use an unbiased clustering to group substances together with a similar mechanism of action, inducing either a lot of protein unfolding or oxidative stress. These two DNA damaging agents induce the p53 response. So in this way, you can group substances together. And over the years, we've built a large database. This is just a small subset of that, where we can now start to compare unknown substances with a database of reference compounds to see what kind of reference compounds give the same response as this new unknown chemical. So it really helps you to identify the toxicological response that is induced by a compound. So in a number of cases, we can actually use these induction profiles, not only to group substances together with a similar toxicological effect, but also to link them to an in vivo outcome, to a pathology, where we see that some of these patterns are typically seen for compounds that either induce toxicity, so neural toxicity, toxicity to the liver or to the heart. So some of these profiles are really linked to a pathology. And I would like to show you one example, that is drug-induced liver injury, DILI, that is has been associated with a number of toxicological effects, mostly oxidative stress, protein damage, mitochondrial problems, inflammation, and a number of these processes we can actually capture in this tox profiler assay. So I said, can we use tox profiler to predict liver toxicity? So to do that, we tested 64 compounds that were labeled by FDA as severe DILI, weak DILI compounds, or no liver toxicants. And we ran these 64 compounds in tox profiler, and we looked at the pattern and see whether we could actually use this to predict toxicity to the liver. So on the right, you see all of the 64 compounds that we tested. It's a large panel. There's no time to go into detail. But as you can appreciate, that many of these substances induced clearly a response in tox profiler, inducing a number of the different biomarkers, either the one for protein unfolding, so that's the ER stress response, unfolded protein response. They induce often LC3, so for autophagy, and in many cases, oxidative stress was induced. And these were also indeed, as just one example, for benzbromarone, a severe liver toxicants. Also in this case, we could see a very strong induction of the ER stress response, and also some of the other responses came up. But looking at these profiles just alone, the profiles just by themselves do not provide enough information to classify the substance as a DILI compound. We did notice that a few of the cellular stress responses, primarily autophagy, induction of oxidative stress, and ER stress, were the ones that were highly relevant for these DILI compounds. And so we focused on these 3 different endpoints and said, so what happens if we if we take the exposure level, so Cmax values of our exposure levels, and take that into consideration. And so what we've done is we took the point of departure concentration for these 3 biomarkers and divided it with the Cmax value, so concentration that are seen in blood in most of the cases actually measured, but you can also model this. And if you look at the ratio between these two, it has been described as the so-called BER, or bioactivity exposure ratio. So the combination of the profile for tox profile in combination with exposure, then all of a sudden we could very well see that this ratio really helped to discriminate between the severe liver toxicants and really set it apart from the no DILI concern compounds. So using that ratio as well as this profile, I think the assay is definitely well able to at least give an indication whether the substance will be toxic to the liver. So to summarize, I hope I've been able to show you that RipRod tracker and tox tracker are validated in vitro models for developmental toxicity and genotoxicity prediction. The interlaboratory ring trial for tox tracker has been completed, has been peer reviewed by OECD, and there's now a positive response to start working on a OECD test guideline. And also the first transferability studies for RipRod tracker are currently running to and of course, the qualification according to ICH S5 has been completed as well. Tox Profiler is a valuable tool for early toxicity testing, for read across, and also for DILI prediction. We have experienced that these NAMs are typically ready for screening, but also for regulatory applications. And it's often that additional mechanistic information that is very valuable in early assessment of the safety of compounds. And all of these assays have already been used for regulatory submissions, typically in weight of evidence approaches, according to different ICH guidelines. And with that, I would like to thank you very, very much for your attention and give the floor back to Masashi. Thank you very much.
Thank you both. What the contents. And I think video record is available for the audiences, and probably the slides will also be available upon request, because there's many contents and people are maybe missing. Do you have any questions from audiences? If you have questions, you can raise your hands so that I can promote you to the panelists. Or if you are shy enough, you can just put in a chat box or Q&A box, hopefully in English, because I cannot translate very well. For the time being, waiting for the question from the audiences, I have a couple of questions. So you develop a lot of methods by your company and approach to the authorities, public sectors. Is it difficult for like a single company to approach to the public sector directly? Normally, because in Japan, it's quite difficult for single company to approach to the authority. Normally, we put like a consortium with having your competitors, for example, and having your evaluator, like a pharma company, etc., and make a team to approach to the authority. What is the scheme in Europe or your companies? Maybe question two.
That is exactly the same as in Japan. So we never, obviously, like anyone else, we also sometimes meet with regulators, but all of these activities in validation are performed either by independent consortia, like for instance, for the tox tracker assay, where there's a validation team that was supervised by the Dutch representative of the OECD. For the ICH S5, a lot of the work has been also done in collaboration with HESI, so the Health and Environmental Safety Institute in the US. So it's always driven by consortia. RipRod tracker was also validated in collaboration with Unilever. So it is always through consortia, through collaborations, and never Toxys by on its own reaching out. So it should always be supported by a consortium and group and never by a single company. So that's just exactly the same in Europe or the US as in Japan, I believe.
Sure, thank you, thank you. For my impression, well, Japanese authority may be more, how to say, conservative, rather than accepting new methods, even though the animal-free method is a boom. And is there any like a two phases of like a department in the authorities who are doing like a conventional method and also accepting new type of method? I heard something like a 3D models for like a organ on chip. In the FDA, there's a more like a active team to evaluate, and they are more prepared to accept from the public sector, no, like the private sectors, to get new ideas. Is there like a two phases of people in the European authority as well?
I'm not sure whether there's two phases, but obviously I think across the world, regulatory agencies are looking at how can we use these new methods to either improve the safety assessment, to reduce timelines and costs, and also to get around animal tests, because obviously there's many examples where the animals are not the best model to predict the effects in humans either. So organ on a chip, organoids, human stem cell based models. I think they're all being evaluated. There is, I think, commissions or groups within the European Commission, at FDA, at EPA. And I think also even in Japan, where JECFAM probably plays a role in validation of new methods. So I think it's not so much different. Sure, every country and every industry has its own, let's say, need to adopt new models. But ultimately, I think it's a global effort to see if we can use also the new technology and you basically use the science to progress the field. And yeah, but the needs are different depending a bit on the regulations. It's ultimately the regulations that drive the innovation, to be honest.
Interesting. You said like timeline. So how long does it take for if you propose new method and they did like a POC and fully file and do the public announcement? How long does it like a
Yeah, so when ultimately I think the best way to get adoption of new methods is to get a formal validation approval through the OECD. Ultimately, it's those are the guidelines that are typically used also by regulatory agencies.
Yep.
For ToxTracker, this validation started in 2017, so that is now ongoing. What is it, 7-8 years? And I think we will need maybe 1 or 2 years to get to a guideline, hopefully. And that is then relatively fast. There's also other assays that definitely take longer. So in general, it definitely takes about 10 years to go through such a validation and acceptance. And there's currently activities going on also at OECD to see how we should change the validation process. Because at this speed, of course, there's no way that we can ever going to get enough of these new models in place. So there's an update currently ongoing for OECD guidance document 34 that describes how to validate new methods, how to see how we can improve that. But I do think personally that validation is very, very important. So it should never be skipped. It's an essential part of the getting the assays ready to be used.
Cool, thank you. I think the duration time frame is not much different to the European, Japanese, like a 10 years, the studies. Okay, thank you. Still waiting for the question, single question from audiences, but if I can continue, if you don't mind. If a Japanese companies or researchers who are normally from the audiences want to sell new compound in Europe, how would you help?
I mean, we're not really selling compounds, to be honest. So that part the companies have to do by themselves. But what we have been working with companies at different stages of product development. So it's either at an early stage where pharmaceutical companies need to select or optimize their lead compounds. for cosmetics let's say to select the ingredients, and maybe even for chemicals to look at different chemical structures and their adverse properties. So that's in the earlier stage, where we basically provide information to help them to develop the substances. And in the second part, where we really try to help is at a later stage when the companies are building their safety dossier, where we can provide information from validated assays to build their safety dossier to show that the substances are not genotoxic, that they do not cause any developmental toxicity. So then we provide, let's say, the data that can be submitted into a regulatory file. Obviously, there are a number of assays that are obligatory that you have to perform, and some of them we don't do. So if we cannot, we are not a one-stop shop in that sense, but we do provide information that no one else can provide in the world. So in that sense, we can definitely support that and help companies, and we have been doing that.
That's great. I also heard your report is very good, and it's not just a issuing report, but like a follow-up afterwards is quite professional. Yeah, I got one question from Fujiwara-san audience. Thank you for the introduction. The great product. Will Tox Profiler be listed in our ECD test guideline for detecting DILI? That's a question.
Well, toxic profiler will be used for DILI testing. Will there be a guideline for that?
That is a personally, I mean, the honest answer is I don't know at this moment, but personally, I think the answer will be no. We really see Tox Profiler as a toxicity profiling assay, and it helps to identify these toxicities. Ultimately, I think if you really want to predict DILI, you need to have a more relevant model. And this goes back to the start of my presentation where it's about fit for purpose. I would always look for toxicity to the liver in a liver model, either in a liver organoid or in a 3D liver model. But these are expensive. The reproducibility is sometimes not there. And in an earlier stage, you would already like to get an indication, does my chemical have a high probability to cause toxicity to the liver? And in that area, I think Tox Profiler can be a very valuable tool. So it's more of a screening tool than a regulatory, let's say, assay for liver toxicity. So there I would simply suggest to use a complex 3D liver model rather than a cell line.
Great, thank you. I think the answer is helpful. I got another question, and the time is limited. So that got my final question. When you carry out stem cell-based assays, do you use one stem cell line or use several lines? Are they human ES cells or iPS cells?
Yeah, so that's a good question. So for ToxTracker, these are mouse cells. It's one cell line that has been genetically engineered, so there's no way we can get around it. In the past, we have validated these cells, whether they represent the effects that we see in other cells or in animals. For the ReproTracker assay, we use human-induced pluripotent stem cells. In most cases, we use one single cell line also to maintain the reproducibility, but the assay has been also validated in other models, and we're currently now setting up a process where we do a second cell line in parallel. So often we use only one, but during the validation we have set up different iPS cells to rule out, let's say, cell line specific effects.
Sure, thank you very much. And time runs quite quickly. And now 5 p.m., 10 p.m., 10 a.m. in Europe, 9 a.m. in the UK. So I think that this session is quite useful. And your feedback from the audience is quite important. And maybe I will send the email for the follow-up email. And if you have any questions or want to have direct approach to Toxys members, I'm happy to coordinate. And for the time being, thank you very much for Gil and Paula for your session.
Yeah.
Arigato.
Arigato. Thank you.
Arigato.
Bye-bye.
Thank you very
Thank you.
much.
Bye-bye.
Bye-bye.
Thank you.
Have a nice
Bye.
day. Bye-bye.
Thank you. Bye. Bye.